So today we're going to continue talking about antibody diversity, and we're going to go into detail on VDJ recombination and how this process occurs. So I showed you this last time to talk about some of the important proteins of VDJ recombination. Today we're going to see a lot of information about RAG1 and RAG2, and so you'll notice those a bunch throughout the day today. We're going to talk about TDT, or terminal deoxynucleotide transferase, and we're going to talk a bit about Artemis today. I will mention very briefly sort of that there are DNA binding proteins involved in DNA repair. Those are listed here, like Q70, Q80, and DNA PK, as well as I'm just going to mention like ligase and polymerase. They are, so as you can see, it is an unusual polymerase. It is DNA ligase 4. Those specific details don't matter. I'm not going to go into the specifics of the exact polymerase there or the exact ligase. So we are going to talk about this process of VDJ recombination that we went through big picture on Friday. If you recall, this cell is going to join a V region and a J region. In this case, we have a light chain, so we just have a V and a J, and paste them together, as well as remove the intervening DNA sequence. So if you were to think about this process, there would be a few things you might guess would be required. So what things, just taking a step back and looking at this, do you assume are going to be required to make this process work? Emilio mentioned one last time. Oh, and by the way, I was so happy with the questions last time. I was in a good mood for the rest of the whole day because I was so happy with the questions you guys asked, so please keep it up. So what kind of things are required? Yeah, Malik. So a nuclease, you need something to do the cutting. You need some kind of enzyme that's going to cut. Okay, what else do you think you're going to need to make this work? Yep, Emilio. A signal sequence. You're going to need some mark that says this is the place to cut. And then anything else that you're definitely going to need? A ligase, so that you can paste things back together. So those are going to be some of the key things that we're going to see today in this process. And the first thing that I need to talk a little bit about is that signal sequence, is the mark that is present that says this is a V segment, this is a J segment, this is a D segment, so we should be cutting DNA here. The signal sequence is known as a recombination signal sequence, or an RSS. RSSs have 3 different parts. First, they have a conserved 7 base pair sequence known as the heptamer. And so the sequence of the heptamer is CACTGTG, which you can definitely sing to Twinkle Twinkle Little Star, which I've never done because I definitely don't have too much time on my hands. So there's always a heptamer with that, and the heptamer always has that conserved sequence. What you can see is that the heptamer is always directly next to the mini gene that we're going to use. So here is the J with the heptamer right next to it. Here is a V with the heptamer right next to it. Then there is a spacer section. And the spacer sequence doesn't really matter, but the length matters, the number of base pairs. And there are 2 different kinds of spacers. One kind has 12 base pairs. One kind has 23 base pairs. And so we have some RSSs that have a 12 base pair spacer, some that have a 23 base pair spacer. On the other side of the spacer is a nonamer. This is a conserved sequence of 9 base pairs, which you can again see here. That will be on the other side of our spacer. In basically every time that you see this drawn out, you will generally see a recombination signal sequence, certainly whenever I draw it, as a triangle. And so you can see that triangle here. That triangle represents the heptamer, the spacer, and the nonomer. And so sort of the big side of the triangle, which is touching the J segment here, would be the heptamer, and then there's the nonomer. And you can also see that we depict these spacers as being different colors. Usually if I'm drawing it for you, I'll shade one in and leave one empty. And so there's some way of depicting the 12 base pair spacer and the 23 base pair spacer as different, or the 12 RSS and the 23 RSS as being different. If you look at the numbers 12 and 23, what do you notice about them? It's not perfect math. It's close to perfect math, though. Yeah, Akuya? Yeah. 23 is almost double 12. Not quite perfect, but it's close. And in fact, what we know is that 12 base pairs is one turn around the helix of normally made DNA. 23 base pairs is 2 turns. And so every so often you'll see these referred to as a 1-turn spacer and a 2-turn spacer. The idea being that you have the heptamer, 1 turn of the helix, and then the nonomer, or the heptamer, 2 turns of the helix, and then the nonomer. So you could imagine that the enzymes are going to be able to grab onto those with appropriate spacing to move forward. These spacers are incredibly important as marks for telling our enzymes where to start this process. They're also important in the regulation of this process. Because there is something known as the 12-23 rule that happens during VDJ recombination. So when we are performing VDJ recombination, you can only ever join a segment with a 12 to a segment with a 23. You can't put a 12 together with a 12. You can't put a 23 together with a 23. You always, when you do VDJ recombination, you're going to be joining 2 segments together. One of them has to have a 12 base pair spacer. One has to have a 23 base pair spacer. I mentioned before that there are 2 different kinds of light chains, either kappa or lambda, when we're talking about the antibody gene production. And you can see they both have a V and a J. Which one has the 12 and which one has the 23 is different between those 2. But the key thing is one has a 12, one has a 23, so you can put them together. With the heavy chain, and this is true of the heavy chain in B cells, the D segments have a 12 on either end. The J has a 23, the V has a 23. And so one thing that you can notice here is that we could not have recombination occur where we put a V and a J together in the heavy chain and skipped D, because V and J have 23s. They have the same kind of RSS, so they can't go together. You can only put them each with a D. You can see another version of this from your textbook here. And so what you can notice is that, for example, at the top with our V cluster, each one of our Vs has a RSS on it. Each of the J's have an RSS, or each of our V's, each of our D's, each of our J's. So we're not going to be able to put a D with another D, or a J with a J, or a V with a J, a V with a V. We can only have V, then D, then J. Here we can only have V, then J, recombining together because of the 12-23 rule. So that's an important part of the regulation of this process, which all comes in from the requirement for the 12 base pair RSS and the 23 base pair RSS. The other thing that I want to point out here is that when we perform this VDJ recombination process, usually the DNA that is between those RSSs gets deleted. So what you can see is that the RSS is actually here both as a orange segment, a spacer, and a purple. Here it's the orange, the spacer, and the purple. Over here, you can see it as well. We're going to have our 23 and our 12 recombine with one another, and the intervening sequence is actually deleted. And so you can see we put V1 with this J, we deleted the intervening V. We've joined the two gene segments together without the RSSs. So from the very beginning of the heptamer is where we start taking out the DNA. You'll see why that is in a second. And we delete that intervening DNA. This is the way we're always going to talk about VDJ recombination. And part of this happens because our protein complex is going to grab on to the RSS with the 12 base pair spacer and the RSS with the 23 base pair spacer and put them right next to each other to start this process of recombination. And the intervening DNA is going to get cut off. One thing that you might notice is in this figure, as well as in the previous figures, you see the RSSs, the 12 versus the 23 base pair RSSs, as triangles that point in opposite directions. There are situations where the RSSs are backwards. Every so often there will be a weird V or a weird J or a weird D that has its RSS backwards. And so if we were to depict it with triangles, the triangles would be facing in the same direction. Like you might have one of these facing the other way. If that happens, and here you can see at the top, we've got our V and our J with RSSs pointing in the same direction instead of in different directions. When we line up those RSSs, the intervening DNA is not cut, and instead it is inverted. It's flipped over. This is what happens in the minority of cases. So this is not something that happens very regularly. I don't want you to focus on this, but sometimes people like to use this as a trick in experiments. And so it would not surprise me if sometimes you saw this when you were reading a paper in experiments. So for example, in this case, the GPT gene is not going the same direction as the promoter, so it can't be transcribed, and we can have VDJ recombination flip it over. And so sometimes people will use this experimentally. Some students will come across this idea of inversional joining from time to time, but it is sort of the minority way that VDJ recombination works. So if you were wondering, well, what if the triangles were pointing in the same direction as opposed to towards each other in the opposite directions, this is in fact what would occur. But we need to talk about the mechanism of this process. And so we're going to go through that in detail. Your textbook has two big figures that show the whole process. One of them is 612, the other is 611. I'm mostly going to be walking us through figure 612 today because it has the level of detail that I want. Every so often I might switch back and forth, but they both are showing pretty similar stuff. And so to start with, we need proteins that are going to bind to our RSSs and recognize them. This is happening at two places with the, in this case, V and J that are going to be joined. Those proteins are going to bind, and specifically those proteins are known as RAG1 and RAG2. So you can see RAG1 and RAG2 recognizing the RSS here. They're actually going to bend the DNA so that they're able to hold on to both RSSs that are getting recombined here. One thing I want to point out right now, if you look at this complex, we have the V segment with a top strand and a bottom strand, and we have the J segment with a top strand and a bottom strand. So there are 4 DNA strands. And in fact, we are going to talk about a process that is happening once by the V and once by the J. But if we drew this out happening twice, if we drew out like the action of all 4 strands all the time, it would get really, really, really messy. And so we generally only draw out one for simplicity's sake, but it is really happening twice. So you can see on the next part of this, it's just showing you an event happening at V. That event is also happening at J. We're just only showing it once for simplicity's sake. Because at some points, the other section shows back up again, and everyone gets lost about where that other section came from. So it's just not being shown here to make your life a little easier. So RAG1 and RAG2 are going to recognize the RSSs. They're going to line them up. And then RAG1 and RAG2 are going to perform a very specific type of enzymatic event, which is called a DNA nick. A DNA nick means that one strand is cut, but the other is not. And you can see that DNA nick here. There's too many R's in this. I have to think very carefully about the difference between our and R. The RAG1 and RAG2 enzymes perform that nick at the border between the heptamer and the V or the D or the J that we're working with. So we have a single strand nick, one strand is cut right between the heptamer and the actual V segment that we're going to be using here. You can see that same nick happening here. When the nick occurs, it looks something like what you see here chemically. We've broken the phosphodiester backbone. So we have the free phosphate on this side of the nick. We have the free 3' hydroxyl on this side of the nick. And you can see that the other DNA strand is intact. This 3' hydroxyl is now going to perform a nucleophilic attack. It is going to perform a nucleophilic attack on the phosphate across from it on the other strand. And so you can see that attack happening here where the 3' hydroxyl attacks the phosphate on the other strand, as well as the end result. So now we have 2 broken strands. One of them has that phosphate and the hydroxyl. This is the heptamer. This yellow is all the heptamer. It's all that a blow up of that yellow triangle. So the heptamer has this phosphate, it has this hydroxyl. The V segment, or the D or the J, the segment that we're trying to work with, you can see the top strand actually is attached to the bottom strand now. The DNA is in a formation known as a DNA hairpin. So we have a hairpin formed with that V, D, or J, and we have this other part formed with the heptamer. The hairpin is on the V, D, or J that we are eventually going to use. It is going to eventually code for our antibody heavy or light chain. And because we are going to use it and it codes for the antibody, we sometimes refer to it as part of the coding joint, or as the coding joint, because it's going to code for something later. The rest of it is the thing we're going to throw away. The thing we're going to throw away contains the recombination signal sequence. And as a result, we often call that the signal joint, because it has the signal in it. So we have these 2 different pieces now, where we've broken our DNA. We have a coding joint, and we have a signal joint that we need to deal with. The signal joint is relatively straightforward to deal with, and so we're going to deal with the signal joint first, just so that we have it out of the way. So there are really 2 signal joints that are made. There's a signal joint at the V, there's a signal joint at the J. You can see that at the top from Param. You can see that at the bottom from your Kubi book, that there's a signal joint on this end by the V. There's a signal joint on this end by the J. from the two things that are made. They're two nice pieces of DNA that have nice ends. One, they have the phosphate and a hydroxyl and look like normal old DNA that you have. So the binding protein complex just takes them and shoves them together, ligates them. We get this nice circle. We all live happily ever after with the signal joint. So the signal joints, once they're formed, are just simply ligated together, and they're done. We'll get this nice circle of DNA that will probably be lost as that cell divides, since it doesn't have an origin of replication. The other thing that this figure shows you about the coding joint, or sorry, the signal joint, is not going to be super meaningful to you until later. But your textbook does show you this DNA sequence example of the coding joint. And if you look at the before and you looked at the after, it would look exactly the same. So you have no change in any of the base pairs with the coding joint. Or as it says here, there is precise joining, it says somewhere. So the coding joint, you just stick it back together and it's done. The coding joint has some problems. The coding joint is much more complicated. And these complications are a good thing. I mean, they're not a great thing for you as a student, but they're a good thing for you as a person with an immune system that is diverse. Because these complications allow for junctional diversity to occur. This is how we get the changes in base pairs, the changes in sequence, that can give us the additional diversity that we talked about on Friday. So as we're putting together our V and our J in this point, VK21 and JK1, we get changes to the specific sequence at the signal joint. So at the coding joint. I'm having a lot of trouble with that today. I apologize for that. So our coding joint is going to have some imprecision, and that's going to give us additional diversity. When we last left our coding joint, it was in a hairpin. So our signal end, our signal joint, was regular old double-stranded DNA. It looked like a regular old double-strand DNA break. You just ligated it back together and lived happily ever after. But the coding joint is in this weird hairpin. And so something has to happen with that hairpin. You can't take the V hairpin and the J hairpin and ligate them together because they're hairpins. They're not double-strand breaks. That doesn't really work. You somehow actually have to break the hairpin before you are able to treat these as double-strand breaks and join things together. And if we were back in my undergraduate immunology class, we would say, and then the hairpin opens magically. But in the time since then, the enzyme that opens the hairpin has been identified. So if you are unable to do VDJ recombination, if you have a problem with this, if you are missing the enzymes RAG1 and RAG2, for example, you don't get an adaptive immune system. You have no adaptive immune system form, and you're probably going to be really sick as a kid because you have no B cells and T cells. And so this enzyme was first found. It was basically a group of kids who had serious immunodeficiencies, were tested to see what mutation they had, and they all had this mutation in a specific enzyme. And someone decided to name this enzyme and say, this enzyme protects kids from immunodeficiency. So I am going to name it after a Greek goddess who is known as the protector of women and children. And thus, this enzyme was named Artemis, who is the Greek goddess of protecting women and children, because the enzyme protects you from having immunodeficiency. And Artemis's job is to cut open this hairpin. So Artemis is pretty great in that sense, in that it is able to cut open the hairpin. However, Artemis has a flaw. If you look at this hairpin, which you can see here, and I'm going to draw some sequence on the board in a second. This bond between the T and the C is a phosphodiester bond, DNA backbone. This bond between the A and the G is a DNA bond, phosphodiester backbone. This bond between the C and the G is a phosphodiester backbone, normal DNA bond. And in fact, Artemis can't tell the difference between them. They all, as far as Artemis is concerned, look identical. Artemis knows that it needs to cut a phosphodiester bond in this region, but Artemis is not able to necessarily know, in quotes, because it's an enzyme, it doesn't have a brain. It does not necessarily know this is the original bond that needs to get broken. Artemis is able to cut basically anywhere 5 base pairs away in either direction. So up to 5 base pairs too early, 5 base pairs too late. We get a cut from Artemis. So it's because Artemis is going to be acting in that location. It's part of a protein complex that's being held right there, but it's not going to cut the exact bond necessarily. If it happens to be lucky and cuts perfectly, then we get the outcome that's shown in the middle, where we have a hairpin, we cut the hairpin open, and now we have regular double-stranded DNA. But if Artemis cuts at point 1 near the top, then we're going to have an overhang. So you can imagine this was originally up here, and now you can see that the DNA is not even. The bottom strand is a little longer than the top. If we cut at point 3, that's shown up there, you can see now we're going to have a top overhang. The top strand is a little longer because we cut here. This is the first way that junctional diversity occurs, is that we can get addition of new base pair sequence from this type of process. And the base pairs that are added because of Artemis's cutting are known as P nucleotides. So what you can see is that we'll have cutting, we might get some kind of overhang, and then some polymerase that's involved in DNA repair will actually fix that overhang to make double-stranded DNA. One of the things that might help you understand P nucleotides is if you know what the P stands for. The P in P nucleotides stands for palindromic. These nucleotides read the same forwards and backwards. And I hope that my example that I'm going to show you on the next few slides makes that more clear. So this is an example of some sequence. You can see that we have a V region, GAG, CAT, AT. And then we have our heptamer, our spacer, and our nonomer. We've got our J region, and then our heptamer, our spacer, and our nonomer coming together to undergo recombination. We're going to get a single-stranded nick in the top strand, whereas the bottom strand is going to stay intact. And so it will look like this. Okay, so we're going to have this. We're going to have the rest of our RSS here. The 3' hydroxyl on this T is going to attack the phosphate on this A so that we're going to get a hairpin formed. So we will have a bond there. And then we're going to have 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. A whole bunch of similar bonds between all of the other base pairs. And so this is what we're going to get. this would be where we just left off. If I could draw on that slide, you'd see a little hairpin between those two pieces. Now let's imagine that Artemis cuts here between this C in the bottom strand and this G in the bottom strand. If that happens, if we just sort of straighten out this DNA, the A is going to be here, this A. This T is going to be here, this A is going to be here, then T, then G. So it's going to look like that. So that's what we're going to end up with when we've opened that hairpin. Does that make sense? Now if you look carefully, you can see that, did I do that right? We can fill this in. And if we box this area, you can see that the top strand reads C-A-T-A-T-A-T-A-T-G when we go from 5' to 3'. And when we go from 5' to 3' on the other strand, it goes C-A-T-A-T-A-T-A-T-G, which is the same. It's the exact same thing forwards and backwards. That's what the word palindrome means in like English and in thinking about words. And so these new base pairs are palindromic nucleotides, or P nucleotides. If we look at the DNA sequence, the original sequence that you find in the genome is this that I've circled. And we have added 5 additional base pairs on top of what is found in the genome. So if we looked in non-immune cells, for example, and we sequenced the genome, we would find this original sequence. We would find that in all cells. But this particular developing B cell has 5 extra base pairs that have been added here because of the way Artemis cut. So that's the first way that we get junctional diversity, is that we have the addition of N nucleotides as a result of how Artemis cuts. You can see that having happened at the top here, and I have edited this figure a little bit from figure 6-12 in your textbook because slightly different events happen on the heavy chain and the light chain. I'm not going to focus on that, but 6-12 does, so I just edited that part out. And so what you can see is another thing that can happen is we can have an exonuclease cleave off some base pairs. So we can actually lose base pairs here, theoretically. That loss of base pairs always happens after the P nucleotide part. And finally, and again, this is happening in a specific order, we have some other nucleotides added. These nucleotides are really different than most nucleotides you've ever seen because they are added in a non-template dependent fashion. They're added by a special polymerase. So whenever you've seen DNA synthesis before, you know how you need a primer so that you have something to complement, or you need a template so that you have something to complement? You need a template. This polymerase is able to add non-templated base pairs. So it just picks whatever base pairs it wants. It just says, I would like a C, and then I would like a T, and then I would like an A. And then that can be complemented. This special polymerase is called TDT, or terminal deoxynucleotidal transferase. And it's able to add these non-templated or random nucleotides. And because they are random nucleotides, they are known as N nucleotides. Once all of this is done, then we're able to ligate our 2 pieces of DNA back together with the normal DNA repair enzymes. TDT is a really nice enzyme. It is used in a lot of types of molecular biology experiments. If you have a piece of DNA and you want to label it in some way, usually you label it by adding in huge excess of label and TDT. And TDT will just take that label and throw it on your DNA for you. It's really quite nice. Yes, Molly?
This is throwing it back a little bit.
Okay.
But when you were talking about how it always cuts right after the T nucleotides, how does that sequence get recognized? Is it just it cuts in palindromic sequences?
It's more that the exonuclease might be present in the enzyme complex. And so that happens to be DNA that's present, free DNA that's present there. That's all. Okay. Other questions? So in overview, this is our coding joint, where we make our hairpins. We bind to these DNA ends. Artemis is going to open them. TDT might add some DNA, and then we will ligate together. But there will likely be some additional new stuff outside of that D and that J, or that V and that J, or that D, whoever. There will likely be this new, besides the original red and the original yellow, there's some new stuff here. This imprecise coding joint that leads to junctional diversity. Yes, Nadia? TDT is also acting as part of the protein complex, and so it's going to be acting in that same location. So I have I'm going to talk about a couple quick things here. So I have a bunch of I have little topics that I have in these slides to talk about, but I also have an old exam question on this stuff, so you can kind of see how we could work through some of it. I want to go through a couple more slides because they are relevant to some questions you've been asking. Then we'll skip to the exam question. Then we'll come back and talk about some of the other material as we have time. So we talked last time about the fact that VDJ recombination has some problems. It is a dangerous process. And we can see many VDJ recombination errors leading to different types of leukemias, lymphomas, or other cancers of the immune system. This is a very dangerous process of cutting and pasting DNA. We definitely see situations where, for example, we have our double-strand break right by our immunoglobulin gene, and we can incorrectly ligate the DNA back together, get a chromosomal translocation that can lead to cancer. These two chromosomal translocations that are shown here are two famous examples of that. So we want to think a lot about how do we make sure that this risky process is regulated? How do we make sure that this risky process does not lead to random cutting of any old DNA to mess up all of your cells? One of the answers is the fact that we have this RSS sequence. And the RSS sequence is the only place where RAGs are going to be able to recognize and cut. Officially, I didn't mention this before, and your textbook actually doesn't go into a ton of detail on this, but RAG1 is the recognizer and RAG2 is the cutter. That will be somewhat important with our paper on Wednesday. One other way that we regulate this process is that all of the enzymes, and you can see this shown in this picture from your textbook with the purple blobs, or this picture from Parham as the blue blobs, all of the enzymes act together. All of those proteins are, in fact, binding to one another as well, and are traveling together and acting together. So once we cut in one place, RAG2 cuts in one place, Artemis isn't going to act somewhere else. Artemis is acting right there in a complex with the RAG enzymes. TDT is there, our ligase is there. All of those proteins are acting together so that the DNA that they are most likely to interact with is the DNA we want them to interact with. They're not going to go off and start interacting with random pieces of DNA. The other big way that this process is regulated is that the RAG2 and RAG1 genes are only expressed at very specific times in the life of immune cells. So in general, when our immune cells are stem cells, they will not make RAG1 or RAG2. They will turn on RAG1 and RAG2 expression when they need to do VDJ recombination. And in fact, at some points in their development, they turn RAG1 and RAG2 back off so that they can undergo cell cycle without risk of damage to the DNA. And then they'll turn it back on. And then once we have this cell finish development and leave the primary lymphoid organ, RAG1 and RAG2 will never be turned on again. And so there's also some very precise control of when we transcribe and translate the RAG2 enzyme, or the RAG1 and RAG2 enzymes, in order to ensure that they are only being made and thus are only present to cut DNA at the time we want to cut DNA. So that's a big part of this regulation as well. Are there other questions here? All right, so I'm going to switch us over to an old exam question to talk through a few pieces of information. If you go on Moodle, you will find this as part of Spring 2017 exam number 1, which is present on Moodle. So first I tell you some stuff about some antibodies. That's earlier. You don't really need to know that to get to this part of the question. And I ask you first that I tell you that there is a human heavy chain that has 3 D segments, 3 J segments, and 3 V segments, and the constant regions for IgA, D, E, G, and M. And then I ask you to draw that sequence. I say to note which RSSs are different, although you don't have to tell me which one's 12 and which one's 23. So, how am I going to go about drawing this? Here, I'll start. I know that the order of these segments is V, then D, then J. In the question, I put them in alphabetical order, not in that actual order. So first of all, there's 3 V's. V1, V2, V3. Then what comes next? D1. Then D2. Then D3. Now I ran out of space, so I'm going to come back over here. Now what comes next? What, Manny? J1. J2. And J3. Okay. So we have a few things here, but a few things are also missing. So we've got our D, our J, and our V segments. But what about the constant regions? Do we know what order they go in? Like a mnemonic for it? Yeah, Arlene. MDs give everyone apples. And so, in fact, you can just draw mu or M, D delta, G gamma, epsilon, alpha. So there's my constant regions. I'm good. I just draw a little box for each one of them. If this was a light chain, we would not have M, D, G, E, and A. Those are not part of the light chain. You could just draw a little box that said constant for the light chain. Realistically, it would either be kappa or lambda, but I'd have to tell you what it was in like the light chain. That's not part of this question right now. So is this the whole answer? Or is there anything missing? The RSSs are missing. All right, so where am I going to draw RSSs? Okay, so let's think about next to the V's first. Where next to the V am I going to draw it? Okay, so it's going to point to the right. Is it going to be on this side or that side? On the right side. So I'm going to draw it here. On that side. Then I'm going to draw another one here. I'm going to draw another one here. Okay. What about other RSSs? Okay, J is going to get RSSs where? On the left? Oh, sorry. All right, J is going to get it on the left. Is it going to be the same color as the V or a different color? This for the J. It's going to be the same kind or the different kinds? This is same. And then what's going to go on the D? One on both sides. And is it going to be the same color or different color? Different color. If I was actually drawing this on an exam, I would have drawn the things in order and wouldn't make it look quite so scribbly, because I would have known that I was drawing them all. So this would be the answer to this question. This is what I'm asking you to draw for here. And asking you to draw this then helps us get to the next part of the question. And the next part of the question asks you, you have further examined antibodies made in this patient and found that those antibodies use V2, J3, and D1. And so then I ask you to draw the final DNA sequence for a B cell that uses those segments. Use the same elements above as necessary. So what am I going to draw here? So what should I start with? Yeah, Molly.
B, D, and J, the correct ones with no space through RSS.
Okay, so I'm going to have, first I'm going to have my V2. Then I'm going to have my D1. Where should D1 be? Yeah, you don't need to necessarily draw the variation here. What I want to see is that they're touching, that you don't have a gap in between them. You don't have them gapped like you had them gapped in the other drawing. Okay, now what do I need to draw? I got J3, right? So this is part of the answer to this question. But there is more to be drawn. Does anybody have any ideas about what else we need to draw here? Yeah, Emilio. All right, we need our constant regions. Okay, there's my constant regions. Anything else I need to draw? I left a space, so probably. Right. So this is the key thing, and this is actually why I wanted to go over this question with you in class, because sometimes I think seeing it this way makes some things click that don't click otherwise. The reactions that we just saw happen with the V, the D, and the J, and at those RSSs in between them. If you notice, V1 hasn't been affected. V1 with its RSS is still there. We didn't do anything on that side of the DNA. If I had given you more than 3 J's, there'd be some more J's over here. But notice that the D's, the rest of the D's, they are gone. We've gotten rid of all of them because we've cut out the intervening sequence on both sides. And so this, in fact, would be the answer to this question. And specifically, I'm looking for you to, have the constant region still there. I'm looking for your VD and your J to be touching. And I'm looking for you to keep the DNA that didn't get deleted. So like that V1 that didn't get touched, I want it to show up exactly the same as it showed up before, because it didn't get affected. Does this make sense? Okay. We're going through this stuff for a reason. No, sometimes this makes more sense than the other one. Different ways make sense for different people, which is why we're doing both ways. All right, we have one other question. And I'm going to tell you something about this question first. You're going to look at this question and you're going to be like, ah! And I always ask a question like this on the VDJ problem set. You will find one just like it. And you will flip out. And then I'm going to explain how to do it, which is what we're going to do now. And then what invariably happens is someone will come to my office and they'll say, Dr. Barker, I just do not know how to do this question. And we'll work through it. And they'll look at me and they'll be like, I totally know how to do this question. I love this question. Can you please ask like 100 of them on the exam? Because I'm going to get them all right. So that's why we're doing it now, is so you can all get this example as well. So you determine that the antibody uses VH2, JH3, DH2. And the following are the actual sequences that you get from the germline, from an unrearranged cell. So that's the sequence of the D segment. I've listed it for you. This is the sequence of the J segment. I've listed it for you. And then if you sequence the DNA in the actual B cell making the antibody, here is the sequence that you get. And I say to label the origins of the nucleotides in the rearranged locus above. If the nucleotides could be P's, you have to label them as P's. Because technically, theoretically, everything could be N. They could all be random. So if it could have been a P, you got to label it as a P. You can't just get away with calling them all N's and moving on from there. People tried. All right, I'm going to draw out this sequence here so that we can label it. All right, so I have CAT. How many cats? 1, 2, 3, 4 cats. ATA. CGG. ATC. GAT. CG. Is that right? There's one more A right here? Okay, is that right? Okay, so also notice this question is worth 4 points. Turns out there are 4 things you got to label here. Each one of them gets a point. So I'm going to label the first 2. And you are going to look at me and say, are you serious that this counts as a point? And the answer is yes, I am. Okay, so the stuff that was originally part of the D is part of the D. That's where it originated. It was part of the D. You can match from the top where I showed you what the D was to the bottom. You can just circle it and say this was the D. And the part that was the J, you can just circle it and say this was the J. And so in the end, you have these 5 base pairs left over. And for each of those 5 base pairs, you have to decide, is this a P nucleotide or is this an N nucleotide? So the way that I do this is I go one by one. If you remember from the slide talking about how these were made, they happened in a specific order. P nucleotides happened, and then potentially any exonucleolytic cleavage, and then N nucleotides. And so we're going to look for them in that same order. We're going to look for P's first, then potentially exonucleolytic cleavage, then N nucleotides. So let's start here with this A. Could this A be a P nucleotide? So I see lots of heads shaking yes. Who wants to explain why? Because in fact it can. Why could this be a P nucleotide? Yeah, Molly.
If you look at it in the context of the whole 5 base pair sequence, you see ATA and then ATA.
Yeah, you actually don't even have to go that far. A could bind to T. So therefore, this could be a P nucleotide. It could have been an A that was bound to that T. And then you just go to the next one. Okay, T. Can this T be a P nucleotide? I see lots of yes head shakes. Who wants to explain why? Yeah, Malik. Yeah, so T could bind to this A. So this one could be a P nucleotide. All right, how about this A? Can this A be a P nucleotide? See, now I see mixed head shakes. What do you think, Marina? Yeah, so A can't bind to C. A could not have been bound to that C. So that one's not a P nucleotide from this side. And as a result, we're done with P nucleotides on this side. Once we stop making them, we stop. It's not like there's not like P nucleotides and then cleavage and then more P nucleotides. Once you know it's done, it's done. Now we're going to think about the other side in P nucleotides. Let's look at this C. Could this C be a P nucleotide on this side? Yeah, this C could have been bound to this G. So this one could be a P nucleotide. What about this A? Can this A be a P nucleotide on this side? Nope, because A doesn't bind to G. So in the end, these have to be N, because they can't be P. The end. And so that would be the full answer to that question. It's one of those things you practice it a couple times and then it's pretty straightforward. Okay, so do we have any further questions about this? All right, so I want to switch back and give you a little bit of additional information. So all of these processes that we have seen thus far are involved in making our antibodies. And this is, of course, happening in a B cell. If you notice, on Wednesday, we're going to be doing a paper discussion. And then after that, we're going to be talking about B cell development and B cell selection. Basically, it's just the timing, the time and the place of VDJ recombination. Now we know the events. We're going to see those events in terms of where they happen and when they happen. But this B cell is going to make a few different things. So one thing that you will notice here is that our B cell has a B cell receptor and also secretes antibodies. And that B cell receptor and those antibodies are in fact basically identical to one another in terms of their binding sites. And so we might wonder, how does the B cell pull this off? The answer is that after the constant region exons, there are a couple of different additional sites. And we can, in fact, put a poly A tail in two different places on this RNA. One of them will end right after our constant region exons and will give us our secreted antibodies. There are also times when we get the poly A tail added a little bit later to that same transcript, such that we actually get a little membrane domain. And so we just put a few extra amino acids on that same protein by simply polyadenylating later, getting a slightly longer transcript. And now we have a transmembrane domain where we originally just had the end to the protein. So we can get basically the same protein as both a secreted protein and a transmembrane protein simply by alternative use of this RNA and alternative polyadenylation. The other question that you might have is about how we deal with all of these different constant regions. How we deal with this, with M versus D versus G versus E versus A. You learned last week that each of those can be really important for different things. You can see those same constant regions here, downstream of our V, D, and J segments on the immunoglobulin loci. If we wonder about class switch and how it's going to happen, it has to do with these exons that make up the constant region and how we use those exons. There are actually two different ways that the isotype can change. If you look back at our chromosome, you will notice that the constant region for mu, or M, is pretty close to the V's, D's, and J's. It's the first one. It's the beginning of MDs give everyone apples. D is the second one, and it's also really close. And then if you look at the other constant regions, they're a long ways away. They're way further in the DNA. So it turns out that there is one mechanism that allows us to have M or D, and a different mechanism that allows us to have these further constant regions. M and D are both made early in the response. They are both seen on B cells that are developing when they're in the bone marrow. Only later, outside of primary lymphoid organs, can we get the second mechanism that allows the use of G, E, or A. Making M and D is relatively straightforward. In fact, when we make our first transcript, that transcript is long enough that it contains both the M constant region exons and the D constant region encoding exons. So we have this transcript that actually, at the beginning, has both M and D on it, and that transcript can be alternatively spliced to either make M and to use the M constant region exons, or D to use the D constant region exons. And so we can have our nice fancy VDJ that binds to our antigen of interest, and then we can just, through alternative splicing of the RNA, alter whether we're going to get IgM or IgD. Notice that with this process, and with that secreted versus membrane-bound process, we were doing alterations of the RNA and the transcript. We were splicing transcripts differently. Those things are reversible. If we don't like a particular RNA transcript, we can have RNA polymerase come and make a new transcript for us. If we change the DNA, it's not reversible. You can't go back and un-VDJ your DNA. But you can make a new transcript and change outcomes of using M versus D or using membrane versus secreted by making a new transcript and newly splicing it. So this is reversible, the other is not. Yep, Marina.
I know that this particular picture shows that the poly A tail is already in it.
Mm-hmm.
Is it sliced and then?
Yeah, so these are sites where polyadenylation could happen. It's actually cut and then polyadenylation can occur. Great point. If we want to have switching to one of those farther away constant regions like G, A, E, then we actually have another type of recombination. We have to cut and paste the DNA again. But this process happens outside of primary lymphoid organs. It happens later in the life of the B cell. So it is something that we are going to really deal with later. But I know some of you who know a lot about biochem get fits from the idea that part of the protein changes and part of it stays the same. And so I wanted to give you, like, yes, there is a mechanism. We're going to have additional recombination where we additionally throw out more DNA to move those far away constant regions close and potentially get rid of M and D. That's going to be how we actually do that isotype switch. But that's a mechanism that we're going to talk more about later. Please make sure to turn in your homework by 5 and be ready to talk about that paper on Wednesday. I will see you guys then.